right, good evening. My name is Zeke, this is Greg, and this is Lauren, and we will be representing Team Savior for this presentation. So, tonight I'm going to go over our introduction as well as our research problem and our research questions, and then I'll wrap up with our survey methodology. Greg will go into a little bit of our genetic work. Lauren is going to talk about our analysis as well as our group dynamics, advice for freshmen, and future plans. So, as you can see above me, the Chesapeake Bay and the Potomac River ecosystems have a wide variety of organisms that inhabit it. So, as you can see here on the food web, the arrows point in the direction that energy flows. So the smaller organisms are consumed by the smaller organisms, but the smaller organisms are consumed by smaller fish and other organisms like bivalves, and then those organisms are consumed by large piscivorous fish, and then eventually by the top-tier predators like the birds in this case. However, the problem that our team has been aiming to address is the invasion of these waters by the northern snakehead, which is a fish native to China that's known for its flat head, its voracious appetite, and its ability to travel very short, muddy distances over land. So as you can see, through our blue arrows that we overlaid on the diagram, the snakehead competes with a lot of the large piscivorous fish, especially bass, for some of the other food that they would normally eat, and it basically has an overlap there in their diets. So related to that, our research questions all fall under one large umbrella question, which is, does the introduction of the invasive snakehead represent a threat to the Potomac River ecosystem? And we will be attempting to answer that question by asking the following three questions. How do anglers perceive the presence of the northern snakehead in the Potomac River? Will the snakehead population expand? And what are appropriate management goals for the snakehead? So we began our investigation into the first question with a survey, which we created last year after getting IRB approval, which is the Institutional Review Board. Essentially, if you have to do any sort of research involving human subjects, even if you're just going to be asking humans questions, you have to go through that process. So we got IRB approval, and we created our survey based on the Maryland Fishery Resource Office's Potomac Creel survey, which is something they do rather regularly. We just altered the questions to fit our purposes a little bit better so that we could make easy comparisons between the two in the future. So we went out and we actually obtained more than 112 surveys this past summer. And because of the format of the survey, we actually interview groups of fisher folk or anglers as they are either putting their boats in the water, preparing to go out on a boating trip, or bringing their boats back in after a fishing trip. So more than the responses of more than 300 individuals were covered in these 112 surveys that we collected all throughout the summer and even into the beginning of October. So we have begun analysis of these surveys, and we've actually found some pretty interesting findings. So a few of these things, a few of the findings we found have been more than 50% of the anglers we interviewed have said that they have caught at least one snakehead over the last 5 years in an area where prior to that there were no snakehead at all. Of those anglers, 95 were able to correctly identify a picture of the snakehead fish that we actually showed to them. It was actually the same picture that we put up on the food web for y'all earlier. We also found that about 40% of those anglers had eaten snakehead, and of those 40%, more than 95% of them would recommend eating it to somebody else, which is some information we will be providing to the organizations we've been working with, as well as, keeping in mind the information I just went over, we found that a large proportion of anglers, especially those whose target species is either largemouth bass or an unspecified bass species, have found a lot less of those species in the Potomac River over the last 5 years. So we've got an increase in snakehead over the last 5 years and a decrease in bass species, which due to the competition, we can draw a positive correlation there, a correlation there. So our next steps for our survey methodology are going to be to complete the summary statistics, such as the ones I've just discussed, finish our a priori hypothesis testing, and integrate our results with the ongoing Maryland Fishery Resources Office's Potomac Creel survey results. Now I'm going to pass it off to Greg, who's going to talk a little bit about the genetic aspect to our project.
So the genetic portion of our project revolves around the principle that genetic variability impacts population size and health. What this basically means is that the more diverse a population is, the better its chances of survival. So, for example, if you had two populations of the same size, but one was more diverse than the other, this more variable population would likely out-persist the other because it could adapt, it could change along with its environment over time. So what Team Savior is doing specifically is that we're going to try and measure the effective population size of the snakeheads in the Potomac River in order to gauge just how variable their population is to help answer our research questions. This all had to start with getting IACUC approval. As Team Attempt said, it is the Institutional Animal Care and Use Committee. And since we're doing genetic work, that means we need DNA, and that means we need samples from the fish. So since we are working with fish, we need the IACUC approval. But this was expedited because the fish we're actually working with have already been euthanized by other parties. So we are not actually harming the fish in any way. So our IACUC process was fairly brief. We started collecting samples in early June at the annual Potomac Snakehead Tournament. This is just a recreational tournament where fisher anglers go out to catch snakehead over 12 hours. And then as they were checking in their snakehead, we clipped fins that we would later use for DNA extraction. And later in the summer, we went electrofishing with Maryland's Department of Natural Resources. They go out every year and electroshock the snakehead population just to try and gauge how the population has changed from year to year. Then we preserved all our fin clips in ethanol so that we could use them this semester, and we have a total of 81 fin clips from the summer work. Earlier this semester, we started working with Dr. David Hawthorne from the entomology department. He was generous enough to give us lab space as well as access to his equipment. Our first step in lab methodology is to extract the DNA from the fin clippings, essentially converting the fin clips to actual concentrated DNA. We then take this DNA and run it through PCR to amplify it so that we can run several trial runs with just one sample of DNA. And then lastly, for the lab methodology, we will analyze the genetic variability using microsatellite markers. Microsatellites are just certain strands of DNA that we have designated and will use as a standard to compare one fish to the next throughout the entire population that we sampled. The primers we are using are from a previously published paper about snakehead, so we know that these primers will work for the species that we are interested in. And through electrophoresis, we'll be able to separate the banding patterns and see just how related these fish are. So far, we have successfully extracted DNA from the fin clippings, and then we ran that DNA through a spectrophotometer to gauge just how high of quality the DNA was. And the DNA proved to be viable enough and of high enough quality that we were able to proceed in our methodology. And since then, most recently, we have successfully scored the microsatellite markers. So we ordered them, got them, and then ran them through electrophoresis, which you can see on the slide above me. The vertical columns are a sample of DNA, and then the horizontal bandings that you see, that indicates the presence of the microsatellite. So those light banding patterns are what we use to see how the fish are related. Our next steps from here are to complete DNA extraction of all the other samples and then run them through PCR to amplify them. Then we will score the presence or the absence of the microsatellites, of which we have 17. So we'll take all 17 microsatellites and compare them with all 81 samples of DNA that we have, and that should give us enough results that we should be able to estimate the genetic variability using a custom-coded MATLAB script that we will make, and OneSamp, which is an open source software. And then all of this just goes back to trying to answer that question of what is the snakehead's effective population size, so that we can answer our second research question of will the population continue to expand in the future. Now I will turn it over to Lauren, who will talk about future plans.
Okay, so hi. Hi. So continuing through the rest of our junior year, we're going to finish analyzing the surveys that we collected over the summer, as well as continue analyzing the fin clips that we obtained over the summer. And we will start to apply for 2 specific conferences that we had in mind. One in Newport this coming April in 2015, and one in Portland, Oregon in August of 2015. Then, as the spring comes along, we will assess and figure out if we need to revise our survey and recomplete it in next summer during the summertime to bring our data to a little bit more focused results that we wanted. And then over the next summer, we could also decide to collect more fish to genetically analyze next fall. Next fall, we will also be hopefully attending our conferences and then finishing the analysis of our surveys and fish, and then writing our thesis to present in the spring. So tonight we have talked to you about our research problem and questions, our surveys and the analysis of the surveys, our lab methodology and what we plan to do with our DNA and how we will analyze it, and our future plans. Our challenges thus far, so our mentor is an off-campus mentor. He works at the Chesapeake Biological Laboratory in Solomons, Maryland. So he's basically an hour and a half away from us when we are working here on campus. So we did not have a lab on campus that we could work in. So we had to search around and find a professor who had the equipment that we needed and was willing to give us space that we could work in. Also, when we were first beginning our project, contacting experts who could help us move forward with our original research was a little tough, because sometimes it was hard as just an undergraduate student to get them to see us as needing help. Also, over the summer, our surveys were about an hour away down in southern Maryland, and it took 3 hours just to survey. So they were really long and time-consuming, and the gas got to be pretty expensive. Also, our project is an off-campus project, so a lot of the times collecting our data forces us to be off-campus and in the field and getting data from that. Also, in our surveys, some of our questions left room for a little bit of ambiguity in the results, so that leaves us some questions where we weren't quite sure how to exactly analyze that data. Challenges in the future that we anticipate, statistically analyzing the microsatellites is going to take the use of our custom MATLAB code. Our custom MATLAB code is going to take a lot of work to make sure that we're getting the data that we want from it, and we're making sure that it's efficient and mimics what the 1SAM open source software will give us. Also, because our team is so interdisciplinary, there are a lot of aspects of our project that not everyone knows everything about. So a lot of us have to go forward and learn about the genetics of the project, or learn about what statistical tests we're doing for the project. So our team composition, we're split into 3 sub-teams. We have a lab team, a statistics team, and the MATLAB team. These 3 teams all work at the same time to bring all of our data and analysis together so that we have our analysis. Also, our team dynamics. So we made it a point when we first became our team to be friends and be really close. So now that we're all really close, we also have a lot of loud personalities on our team. So sometimes during our meetings, it gets pretty loud and almost seems a little bit chaotic, but we get everything done. So we have some advice for freshmen as they enter their own teams. One thing that we really focused on when we were getting started was having a pivot foot. A pivot foot is something that you know is going to work and that's going to get you data, and then that way you can continue moving forward and try something a little bit more novel. Also, you really want to set your internal deadlines for before something is due, so that you know that you will have something done and that everyone is proud of when it comes time. Oh, finally, you want to be flexible, because your project is going to change from its original advice from the beginning. So we would like to acknowledge Dr. Joe Love from the Maryland DNR, Mr. Josh Newhart from U.S. Fish and Wildlife Services, Dr. David Hawthorne, who's helping us with our lab, Mr. Alex Carroll, who is one of our librarians, Ms. Eileen Harrington, who's another one of our librarians, Dr. Tom Miller, our mentor, and the Gemstone staff. Are there any questions?
What are your anticipated results from the DNA extraction part of your research project, and how does that tie back into the surveys you're doing today?
The question was, what are our anticipated results for the genetic portion of our project, and how does that tie back to our overarching research question?
So the snakehead population is fairly young in Maryland. It was first established in 2001, so as of right now, it's still considered to be a relatively small population. So we do expect there to be some fair amount of DNA crossover and them to be fairly related. Regardless of how that turns out, that data will be taken back to seeing how variable the population is, because that alludes to how healthy the population is, how stable is it, are we going to be stuck with them forever for the next 2 years, that sort of thing. And that should be able to help DNR and Department of Natural Resources sort of tweak their management plans accordingly.
If your DNA wasn't of a good enough quality that you could use it, what was your alternative? What was your backup plan to evaluate the diversity of the fish?
So the question was, if our DNA was not viable, what was our backup plan? What was our pivot foot, essentially?
Well, first we would have tried to tweak our methodology. The extraction kit we actually were using was not for fish fin clippings, so we sort of just had to go off the cuff with Dr. Hawthorne to try what we thought would work, and it did, so that was excellent. If that did not work, we would have still tried to figure something else out. We have our surveying, which would have been a fallback regardless, but we would have eventually found some way to extract DNA, because extracting the fish DNA is not all that uncommon of something to do.
How often, especially when you were surveying, how often do they fish, and do you take into account like more regular fishermen versus ones who went out?
So the question was, did we take into account how regularly the fish or fisher folk or anglers went fishing, or for how long they went fishing, that kind of demographic information, correct? So the response to that question is that the first portion of our survey was actually almost entirely demographic data related to fishing habits. So there are questions like, what is your preferred method of fishing? Do you use rod and reel? Do you bow fish? And then there are questions like, how long is your average fishing trip? How long is this trip? And questions like that, so that we would understand how experienced of an angler we were talking to, as opposed to maybe somebody who is really going out for a joyride just trying to catch a catfish or two for dinner.
You mentioned your data might help with like management plans. What kind of like management plans are there out there right now? And do you have any like new ideas about how to manage the population?
The question was, what current management plan is available or in use, and how will our data affect that?
So the current management plan out there right now, the state and federal governments, they put out a lot of tournaments. So we talked about the Potomac River snakehead tournament, and there used to be rewards for catching them, and in the state of Maryland, snakeheads are like kill on sight, so if you catch it and you plan on keeping it, you have to decapitate them. And they also they're trying to put out more publicity about the snakehead. We're hoping that our project will essentially give more information about the snakehead in the Potomac River. So there's not a lot out there on the snakehead in the Potomac River right now. There's a lot of studies that have been done in New York, but we don't have a lot of information for here. So hopefully our information will help guide Maryland DNR to tell them what to do.
So you were discussing
the surveys you did with the anglers and that you were surveying them right before they went on a fishing trip or right after, how accurate can you expect the data to be about how many fish they caught if you're kind of asking them on the spot?
So the question was, we asked anglers with these questions when they were coming either back or going out on a fishing trip. So how accurate would we expect our responses to be?
So instead of asking the anglers, how many fish did you catch today, we asked, what is your target species today? What would you like to catch when you went out? To get maybe an idea of the things that they enjoy catching, maybe something they catch a lot, just to find out different things about just things like that. Thank you.